Okay, I thought we might have a little smaller group today. We're almost half though, so not too bad. Got a good solid group here though, so that's good. Let's jump right in it today. We were finishing up, or we just begun the process of thinking about how microbes infect hosts. So we were talking about a lot of terms. We talked a little bit about our normal microbiota. Remember, our test is a week from today. So by all means, go do something fun over spring break, but use that time maybe to catch up if you need to, get that homework wrapped up so that's not hanging over you. I will send out an email after class today telling you exactly where the cutoff will be for that exam. So hopefully just what we covered today. So let's keep moving. Again, lots of terms last time. Hopefully some of those were a little bit familiar already. Let's talk about the way that we describe the disease progression. There's 5 different periods that we can use to describe this. And so in this graph, you'll see the red line corresponds to the number of pathogens, and the blue line is severity of the signs and the symptoms. So you can see typically when you have an increasing number of pathogens, then that's going to be reflected in the host severity of symptoms. But right at the beginning here, you can see that they don't quite match up. You can, when a host is first infected, it takes a little while for the numbers of pathogens to start really having any effect in the host. So we call that period the incubation period. So here the host, no symptoms, no signs or symptoms. Okay, even though the pathogen is multiplying, the host doesn't have any symptoms yet. When the host begins to have symptoms, we call that the prodromal period. We'll say mild symptoms, and they're usually pretty general symptoms. So, you know, maybe things like, oh, I've got a scratchy throat. Okay, that could be a lot of different things. That could be strep throat, you know, that could turn into the flu, it could turn into a cold. Okay, just kind of a mild, really mild symptoms. A little headache, oh, what's that going to turn into? And then when the illness is at its peak, most severe symptoms, signs and symptoms, we would call that the period of illness. Occasionally, a pathogen might kill the host, and then that's the end of it. But assuming that the immune system starts to kind of win out a little bit, next you go into a period of decline, where again, symptoms are getting a little bit milder, immune system starting to win out, and then convalescence, again, no signs or symptoms in the host. Still could have maybe some kind of chronic damage, but potentially though. It doesn't mean that they, of course, will always return to full health. When is the host contagious? Well, it depends on the infection. The host could be contagious during any one of those 5 stages. It's particularly difficult to control an infectious disease when people are contagious even in the incubation period, because they're out and about, they feel fine, they don't have any symptoms. So of course, when we were infectious with COVID, even during the incubation period, chickenpox is another one that's very infectious in the incubation period. Again, those are hard to control. Sometimes even during convalescence, people can be infectious, particularly with diarrheal disease. They can still be shedding pathogens in the stool and be infectious for quite some time. But more commonly, when people are the sickest is typically when they're most infectious, but that's not always the case. All right, so how pathogens cause disease? And just a step back, kind of a little historical perspective here. Of course, for millennia, we didn't know what microbes were. We thought things like bad air or foul smelling air caused disease. Robert Koch is given a lot of the credit to help us kind of think more scientifically and help us to understand what actually causes disease, especially in terms of infectious disease. Okay, so for any infection that is for any disease that is infectious, Robert Koch, and this is again late 1800s. Remember, he was a contemporary of Louis Pasteur. Yeah, we mentioned him like the first day of class, I think. He came up with these criteria that if they are satisfied, then that's definitive proof that a certain microbe causes an infection. Okay, so and we'll talk about how there's problems with this. This doesn't always work, but this is a really big step forward. Robert Koch was known for his meticulous experimental design. So he said, okay, if you have a healthy organism, then basically that pathogen that you're trying to test for, the organism is healthy, then that pathogen isn't there. Okay, so that's sort of the first premise. So the suspected causative agent has to be absent from all healthy organisms, but any organism that is diseased, you should be able to see that suspected agent under the microscope, as well as be able to grow it up in pure culture in the lab. Okay, so that's the second criteria. You have to be able to grow it up in the lab. Third criterion: if you take that infectious agent and now inject it into a healthy susceptible animal, that animal will get the same infection. And then if we can isolate that causative agent again out of that diseased animal, then that's proof that whatever agent it was that you grew up is the etiological agent or the cause of that particular infection. Okay, so hopefully you might be able to think of some ways that we wouldn't be able to prove certain diseases. Robert Koch was kind of lucky. One of the first agents that he discovered was the causative agent of anthrax, Bacillus anthracis. So that was an organism that was able to be grown up and able to be seen under the microscope. If he would have picked some viral disease, he wouldn't have been able to do that, right? He wouldn't have been able to culture it. There are some other limitations that we could think about. Obviously, there's a lot of diseases that aren't infectious, things that are genetic or maybe caused by a nutritional deficiency, for example, or an autoimmune disease. Those aren't going to have an agent per se that we could grow up. In addition, sometimes pathogens are found in healthy individuals, yet they don't cause disease. So I mentioned last time, I think the causative agent of whooping cough, Bordetella pertussis, sometimes is associated in our kind of upper respiratory tract with healthy adults, and in small numbers. We can still pass that on to someone that's susceptible, like a baby, for instance. Another example, my daughter was diagnosed with mono about a month ago. And she hasn't been around anybody that's had mono, but she said, I kind of remember there's been a few instances where I shared drinking glasses with a group of people, and probably one of those people had that virus kind of, again, associated somehow, with them, and they weren't sick, but they ended up passing it on to her. Okay, so that wouldn't fit that Koch's criteria there. Also, not all test subjects are going to be equally susceptible. So for instance, why would some people get exposed to an infection and not get it? Well, there is some role of our normal microbiota. Okay, everybody has a little bit different constitution of normal microbiota. And definitely, we're going to get more into this in the next few weeks. There are differences in immune response between individuals. Okay, the way we process and present antigen is slightly different between individuals. So, you know, two people can be exposed to the same pathogen, the same numbers, and both be healthy, and one of them gets sick and one of them doesn't. Okay, there's just differences there. And not all pathogens can be cultured. Okay, like viruses, we can't culture viruses in the lab. There are even some bacteria that we haven't been able to culture. We have to maybe grow them in an animal, or we just haven't, we know they're there, we just haven't been able to culture them. We don't have the right media, maybe. Okay, so again, there's problems. It's not perfect. It's a good step forward. In 1988, Stanley Alcow developed kind of a follow-up to Koch's postulates. And he called them the molecular Koch's postulates. So I don't want to talk in too much detail about this, but what the molecular Koch's postulates show here, the goal is to find a gene that causes the organism to be pathogenic. Okay, so the example that they give here, there are strains of E. coli. Like, we all have E. coli in our large intestine that doesn't do us any harm, but there are certain strains of E. coli that have acquired the ability to make a certain kind of toxin, and then that can cause diarrheal disease, different manifestations of diarrheal disease. And so we can, by molecular biology techniques, try to isolate the gene that makes that toxin, pull it out of the organism. Okay, now we see, oh, it's not causing any harm to a test animal, and then we can try to put that gene back in and see if now it does cause the disease. So it's kind of a variation of the original Koch's postulates that they called molecular Koch's postulates. And there's still limitations with this. Sometimes we can't do the gene manipulation. And then sometimes we just don't have a suitable test animal to do that on, but it can give us some interesting information and help us to understand what makes organisms disease causing. Okay, so a little bit more terminology as we move forward here. These two sometimes are easily mixed up, and I'll try to use them correctly. But if we talk about the capacity of microbes to cause disease, that's pathogenicity. Okay, we've been using the word pathogen, a disease-causing microbe. If we talk about, can the microbe, is it pathogenic? Okay, if you do this, if you use that word correctly, that's a yes/no answer. Yes, it's pathogenic. No, it's not pathogenic. And then when we talk about how pathogenic is it, that's really not a correct use of the word. It's best to say, if we're going to talk about the disease-causing ability, then that refers to the virulence of the organism. Okay, so language can be cumbersome, but sometimes I will say this too, like, oh, it's really pathogenic. It's more correct to say that organism is highly virulent. Okay, so when we say it's highly virulent, that means that organism is likely to cause disease, and it's likely to cause a very severe disease if it's highly virulent. Okay, so we'll look at some examples of that. what makes an organism, what gives it the capacity to be pathogenic, or what makes it more virulent, that depends on the virulence factors that an organism possesses. So we're going to look at lots of examples of virulence factors. These are things, if you think about it being a war between the pathogen and the host, virulence factors are kind of like the arsenal of ammunition that a bacteria might have in order to cause infection. So things like toxins, things like proteins that it makes, or things that it does to dampen the host immunity. Those are examples of virulence factors. Okay, so getting back to this term virulence here, if we look at the relative virulence of some microbial pathogens, okay, and again, these are just examples. Don't memorize these examples, but a highly virulent organism, like I said, is one that's likely to cause disease. Okay, whenever it's exposed to the host at the right portal of entry is what we're going to call it. We're going to talk more about that term in a minute. But for instance, if it's, you know, this Francisella tularensis, we talked about that infection briefly last time or the time before, tularemia. That's the one that's carried by the fleas on rabbits. If you inhale that, that's the correct portal of entry for that microbe, then you're really likely to get sick. It's a highly virulent organism. The bacteria, again, has a lot of virulence factors that help it to be successful in establishing itself. And that's a very severe infection that can has some pretty high mortality rate. Yersinia pestis, bubonic plague, another very successful, highly virulent organism. Whooping cough, a little bit less so. These two organisms, Pseudomonas and Clostridium, are only going to cause infection when the conditions are just right. Same thing with Candida. We talked about that as, you know, if a patient is on a broad spectrum antibiotic, then again, the Candida that might normally be there kind of overgrows when other organisms aren't there to keep it in check. And less, really not virulent at all, we might say avirulent, is the kinds of organisms that you would find in yogurt, like lactobacilli. Okay, we ingest those all the time and they don't make us sick. So there is some difference, certainly in pathogens, as to how likely they are to cause an infection. Another way that we measure virulence sometimes is by the infectious dose or the lethal dose. It's two different ways we kind of express how many cells or virion particles, for example, it takes typically to make someone sick. And there's a big variety. Sometimes it might only take one cell. Now, Q fever is infection that's kind of like the flu. It's not particularly severe. Actually, in this case, some infections might take like 1 billion cells. Cholera. We get cholera from drinking infected, contaminated water. So it's really easy to get 1 billion cells when you drink some water. So, but sometimes the idea is sometimes the lower the infectious dose, then the more virulent. Okay, so we don't, in other words, if it takes a whole lot of cells to cause an infection, it's maybe a little bit less virulent. And again, sometimes the problem with this is that individuals are different, right? What the number that causes an infection in one individual might be different from the other. So we try to express this as kind of an average of test animals, either the average number that makes test animal sick, or sometimes it's easier to assess whether it kills the test animal or not. Not hard to judge whether they're sick or not. Okay, so again, organisms with low infectious doses are considered highly virulent. Okay, I wrote that right. All right. So here's a table from your book that's just sort of interesting. Again, don't memorize this, but it has some infectious doses for a number of pathogens. We've talked about a few of these. I think I've mentioned norovirus in class maybe once or twice. A really low infectious dose. That's the infection that's really super contagious, makes you come out at both ends, often at the same time. It's just really, really nasty. Again, low infectious dose. Some of those pathogenic strains of E. coli, really high infectious dose. But again, sometimes only a single cell. So Giardia, that's a diarrheal disease, protozoan diarrheal disease. So again, and sometimes we have no idea. If we don't have a test animal to do this on, then we wouldn't want to test this on humans. Here's an example of an infection that is quite virulent because it only takes a few virus particles, we think, for someone to inhale to make them ill. If one person with this infection walked into a room with 100 people that were susceptible, probably 99 of them would get this. Any guesses? Bright red rash, measles. Right. So this is one of the reasons we have to keep our vaccination rate high, because this will start to spread as soon as that vaccination rate drops in a community. So it used to be before a vaccine, just everybody got it. A few other terms that I use quite a bit, this term opportunistic pathogen. So an opportunistic pathogen is one that will only cause disease in certain circumstances. Where a true pathogen is going to cause disease regardless of the host status. We'll say host health status. Okay, so some examples. A primary pathogen, or what we sometimes call a true pathogen, it doesn't matter who you are, it doesn't matter if you're the picture of health, but a true pathogen could make you sick. Rabies, influenza, anthrax, there's a ton of true pathogens. An opportunistic pathogen is only going to make an individual sick if the conditions are just right. So usually one condition might be if the host immunity is a little bit lower than usual. So what are some of these opportunities? Is the host pregnant? Pregnant women, their immunity is just a little bit lower. That's why we saw COVID having a little bit higher mortality rate among pregnant women. Let's see, what else? Burn victims. They have, if they have lost their first barrier to infection, their skin, they're really susceptible to infections that otherwise wouldn't be able to cause an infection. Maybe a person is on some immunosuppressive drug, something like a steroid, like prednisone, for example, or undergoing chemotherapy. That's known to knock down our immune response. So those are some examples. And sometimes if the pathogen is introduced into an unusual site. Okay, and it may be normal microbiota. Okay, so for instance, you've got lots, you've got E. coli in your colon that's not causing you any harm. It's part of your normal microbiota. What if a patient's appendix bursts or they get stabbed in the colon? perforated bowel. Then they've got those organisms that are normally not causing any harm out in places where they shouldn't be. And then, yeah, now the correct circumstance has arisen, and now the patient's got a huge problem. So, you know, microbes on our skin not causing any problem until we get a wound, maybe, and then they can get into our bloodstream and cause a big problem. So, those are opportunists. All right, so let's look at the different stages. We kind of like to throw a label on a pathogen as it's infecting a host. There's got to be some exposure to the host, and that's going to be at a portal of entry. We're going to look at those different portals first, the ones that are the most successful. That pathogen usually has to stick, be able to adhere to certain kinds of host tissue before it can invade deeper into deeper tissues. And then if it's going to be able to establish an infection, usually it has to have a way to be safe from our host immune cells, our immune response. And then it needs to be able to leave that one host, usually in greater numbers than it came, so that it has a chance of infecting other individuals. All right, so those are the stages that we're going to walk through. If you're looking at an organism that's coming to a host from the outside, then we'll look at the portals of entry. So I'm not talking about an organism that's part of your normal microbiota already. So you're exposed from the outside. Here's the different portals of entry that we see. Skin is not a common portal of entry. As long as it's intact, we have a lot of defenses at our skin. That's not very common. Okay, we're going to look more at those defenses when we get into our immunology section next. So more about that. Again, not common. Probably the most common is portal of entry is going to be our mucous membranes. So this is any place where you have an opening in your body to the outside. So your mouth, your eyes, urinary tract, reproductive tract. Those are our mucous membranes. We have less defenses there. The layers of cells are thinner. One of the main reasons the skin is not a good portal of entry because our cells are dead on the outside and they're continually being lost. So pathogens can't attach there very easily. So again, less defenses at mucous membranes. The most common portal of entry by far here is the respiratory tract. Okay, so our other mucous membranes, we have kind of a flushing action of, fluids that are, like, our urinary tract, that urine flushes out microbes. We have cilia in our respiratory tract, but that's maybe not quite as good. I'm gonna, again, talk more about that. Placenta, not common. There are some pathogens that can penetrate through the placenta, but it's kind of a short list. And then kind of a catch-all, the parenteral route. So I brought that word up last week, but remember that root, enteral, that means intestinal. So par means not the intestinal route. So basically we use that word in this sense, like not the normal route. So if we're exposed by not a normal route. So this is kind of a catch-all for any time you have like a break in the skin. It might be an insect bite. It might be a wound of some kind. It might be a needle poke. Those are all considered the parenteral route. Okay. All right, so once the microbe comes to its correct portal of entry, and microbes are usually pretty specific about that. We could swallow HIV and we wouldn't get sick. It's not the microbes, the viruses portal of entry that it needs. So once they enter the correct portal of entry, they usually have to adhere in some way in order to be able to colonize the host. So if we think of some examples of adhesion factors or specialized structures or proteins that the cell or virus might have to be able to do that. We talked about a few already that bacteria have, pili or fimbriae. Usually we think about, we call these special hair-like projections that are important for attachment, fimbriae. Occasionally, you remember they've got the same protein, whether we call it pili or fimbriae, kind of same idea. And then bacteria are often going to have some kind of attachment proteins. And for bacteria, we call these attachment proteins either ligands or adhesion proteins. Or sorry, adhesins. Adhesions. Whoops, no O there. Adhesins. Sorry, I'm not kind of sloppy. Okay, so here's some graphics to explain that or give you a picture. Okay, so remember the flagella are long, they're for locomotion. Any organism that's got fimbriae, those are really short hair-like things that help the cell adhere. They're going to typically have proteins at the end of the fimbriae that are going to make them specific for a certain kind of tissue. Here's just a drawing of bacterial ligands. Okay, so here's the bacterial cell. Here's the host. Hosts are often going to have some kind of receptor, might be a sugar or like a sugar protein, glycoprotein typically, where the bacterial ligands will recognize. E. coli, for instance, here in this picture, you can see the capsule. That's what that kind of dark stained part is. E. coli also has lots of fimbriae that help it anchor itself into our large intestine. Without those fimbriae, it would definitely get washed through the digestive tract. Okay, so capsule helps make it sticky a little bit. In this picture, I actually decided not to post this, and then I went ahead and did it anyway. But this is supposed to show bacterial cells making a capsule and sticking to some kind of a catheter. Okay, so it's kind of hard to tell what's what in that picture, though. Here's another example. Again, don't worry about these few examples here. I'm just illustrating it. But for COVID, of course, COVID's got these viral spikes that help it adhere to specific receptors that host cell has. And for COVID, it's a ACE2 receptor. You might have heard about that. It stands for angiotensin, now I'm forgetting. Say that louder. Converting enzyme, thank you. Angiotensin converting enzyme. And you might think, why do we even have these receptors? But they help, one of their functions is to regulate the blood pressure of the host. So when COVID latches onto those receptors, then those receptors quit working. So if an individual is already having trouble with high blood pressure, then now the receptors that help modify that aren't working at all because COVID's attached to them. But that's how it tricks the host cells into engulfing them. We talked about that receptor-mediated endocytosis, a way that some viruses gain access to host cells. So that's what COVID does. And one more example, bacterial pathogen, Bordetella pertussis, causative agent of whooping cough. That is a bacteria that actually doesn't invade further into our tissues. It just sticks on the ciliated cells of our respiratory tract. So it's really important that it sticks there, and that organism makes like 8 or 9 different toxins and proteins that help it stick to those cells. So we can have, lots of different ways to adhere to surfaces. Okay, so as we think about a pathogen adhering and now it wants maybe to invade further into tissues, some virulence factors that we're going to take a closer look at, extracellular enzyme. I'm going to show some examples of those. If you're in lab, you had a whole lab on exoenzymes, same thing. Toxins, especially bacteria, make a couple different kinds of toxins that we're going to talk about, as well as anti-phagocytic factors. Okay, so these are kind of the main groups of virulence factors. Again, these are things that make the pathogen more likely to be successful in invading the host. All right, so some examples of extracellular enzymes. Our cells have, you could kind of think about as kind of the glue that helps them stick together. It's called the hyaluron, or sometimes it's called hyaluronic acid. If bacteria can make an enzyme called hyaluronidase, that's an enzyme that's secreted by the bacteria that will help it break down that hyaluron in between cells so that it can invade between cell layers. Okay, so it's a mechanism of invasion. Here's a couple other examples of extracellular enzymes. Coagulase is one. This didn't match up quite right. You're going to test for that next week in lab. Coagulase secreted by certain bacteria. Not very many have the ability to secrete this, but coagulase forms a clot in the blood. Okay, so here's a bacteria. It's forming a clot around it. So it's effectively walling itself off from our host immune cells, like neutrophils and macrophages that would come and engulf those cells and get rid of them. Okay, so they can grow in number. When the time's right, they're ready to, they've got some critical number and they're ready to go out and invade further. Then kinase will break that, dissolve the clot so that it can invade further. Okay, so that's a really effective way helping the organism to establish an infection. Okay, so your book mentions a couple others. We don't worry about the class here, but we talked about hyaluronidase. Collagenase is kind of similar to hyaluronidase. It helps organisms break down collagen, connective tissue, help it invade further. This DNase, I kind of left this up here because this will make more sense a little bit later, but that can help organisms kind of get around some of our host, the neutrophils actually will try to trap bacteria by throwing down this kind of net of DNA. And so if bacteria can make this exoenzyme, DNase, then it can kind of break free of that. This phospholipase C, I'm not as familiar with that one. We'll just skip over that one. I'm going to refer back to that in a second, though, so stay tuned. A couple different kinds of toxins. Again, especially bacteria. I don't know if there are any viruses that have the ability to make toxins. This is pretty much a bacterial thing. And there's two main types. We've talked about one of them already. We've talked about endotoxin. Remember, endotoxin is lipid A, which is parts of the lipopolysaccharide of gram-negative organisms only, okay? Because that's part, I'll come back to this slide in a minute. That's part of the outer membrane of bacterial cell walls, okay? That lipid A is embedded right in that outer membrane. So it's only released when that gram-negative cell dies. Anytime that membrane is going to be disrupted, mostly when it dies, that will start to break down. So when it's phagocytized or when we treat a gram-negative infection with antibiotics, kind of a big die off at once, a lot of endotoxin release all at once. So it's a small effect, small part when the cell divides, that cell outer membrane might be disrupted a little bit. Okay, so anytime the cell wall is disrupted, So we have to be careful when we give an antibiotic to kill a gram-negative infection. We have to give lots of anti-inflammatories as well to kind of tamp down the result of that endotoxin. Whoops. So the effects of endotoxin, low toxicity, so it causes things like fever and inflammation. We're going to talk a lot more about those things coming up when we talk about our innate immunity. Sometimes inappropriate blood clotting. Okay, and other kind of non-specific general effects. Again, often not a problem unless you have a huge infection and all those cells die at once, then that low toxicity now becomes a problem and can become life-threatening to the host. So the other kind of toxin, exotoxins, they are proteins, okay? And this can be gram-positive or gram-negatives, both. And these are secreted proteins. So what's the difference between an exotoxin and an extracellular enzyme? An extracellular enzyme is usually only going to work right around the cell that it secretes it. Okay, so extracellular enzymes, they are going to act locally. Sorry to flip back and forth on you there. But an exotoxin is going to act more systemically. And what I mean by systemically is around the entire body of the host, or certainly into many different tissues or organs. Okay. Now, I did want to, when the book gives certain examples of exotoxins, and one of the examples that it gives is this phospholipase C. It says it calls it both an extracellular enzyme and a toxin. So apparently there's some gray area. Like, well, maybe this is something that secretes a little bit further from the cell, but not quite systemically. So I don't really know the story of that, but I say don't worry about it. I probably shouldn't have even brought it up. Exotoxins are considered to have very high toxicity. So it only takes a little tiny bit of it to have an effect on the host. Botulism toxin, a 6-ounce bottle is enough to kill everybody on the planet. Okay, really toxic. So when we use botulism toxin for therapeutic things like to control muscle spasms or to control wrinkles, we dilute it millions and millions of times so that it doesn't kill us. Okay. Let's see, what was I gonna say there about it? They're also very specific. Okay, so they act on a particular kind of cell, most likely. So here's some examples of exotoxins. Cytotoxin, something like a cytotoxin that just kind of acts to kill cells. That's kind of a general term. A neurotoxin will specifically act on nerve cells. Botulism toxin is actually a neurotoxin, along with tetanus toxin. Those are infections that we might get to talk about at the end of the semester if we have time. Something that's an enterotoxin, okay, not an endotoxin. Entero, remember, enterointestinal is going to affect the cells lining the gastrointestinal tract. So an organism that causes some kind of food poisoning is probably going to secrete an enterotoxin. It's going to cause increased motility of bowel, going to cause diarrhea. Okay, so those are some examples. Oh, and then, yeah, your book, this is just a nice table comparing endotoxins with exotoxins. So again, these can be helpful to kind of cover it up, see if you've got the basic idea. Don't worry about the heat stability. It's one detail we just won't worry about. And then some examples of anti-phagocytic factors. We have talked about phagocytosis already, and we will talk about it in more detail, but remember, a phagocyte is going to be a white blood cell that engulfs and removes invading pathogens. So remember, our cells don't have a cell wall. Okay, so some of our cells can be phagocytic. They can extend pseudopods, engulf a bacteria in an endosome, a membrane, which we might call a phagosome. And then the lysosome is going to fuse with that phagosome. Remember, within that lysosome, lots of digestive enzymes. We'll talk more about what's in there later. But when the lysosome fuses with the phagosome, then we call it a phagolysosome. Most of the time, 99 times out of 100, that's going to kill the invading pathogen, and it's going to be released outside of the cell. Okay, so that's the basics of phagocytosis. Unfortunately, pathogens have ways around that, both keep from being engulfed in the first place, and some pathogens can even survive inside and maybe keep the lysosome from fusing with the phagosome, for example. So some mechanisms to prevent engulfment. We talked about capsule production, how that's, for instance, that organism that Frederick Griffith worked with, Streptococcus pneumonia, was not pathogenic if it didn't have the ability to make a capsule. Okay, that capsule keeps it from being phagocytized. Now, our immune system, though, that might keep our innate immune cells like macrophages and neutrophils from engulfing a pathogen, but eventually we can mount a very specific result and response and make antibodies that might be able to recognize that capsule. So that's what part B here is showing. An antibody that has bound, here's the antibody that's bound to a bacterial capsule. Our phagocytic cells have receptors for one end of the antibody. This is a little bit kind of a confusing picture because the cell, the bacterial cell and the antibody are drawn so huge that it's hard to picture that cell engulfing it. But basically, those antibodies, give our phagocytic cells kind of a handle to hold on to. So now they can be phagocytized. There are pathogens, though, that produce proteases to degrade enzymes, to degrade antibodies. Okay, so it's this arms race, like, oh, well, we make antibodies. Well, then they destroy the antibodies. Okay, to make them, again, the pathogens more likely to not be engulfed. So a few other examples. M protein is kind of like the fuzz on a tennis ball. So here's a cell that's real fuzzy on the outside. It's a Streptococcus pyogenes. That's an organism that causes some things like strep throat, some other more serious infections. And here's a cell that doesn't have that M protein. The cell that doesn't have M protein doesn't survive in our bloodstream at all. But if it has M protein, it helps the organism resist phagocytosis. And also some other things like complement that we'll get to later. So other things that would help the cell be more likely to get phagocytized. That's just, again, an example of an anti-phagocytic factor on one specific organism. And there are this is kind of related. There are some organisms that are really good at varying their surface proteins. So, and that will affect whether it's likely to be phagocytized or not. So a couple of examples, the organism, Lyme disease is the one that your book talks about. Borrelia burgdorferi is the bacteria that causes Lyme disease. It's really good at changing its proteins on the surface. So the antibodies that we make once we make them, they're not good anymore. And then it takes another week or so for us to make more antibodies. So Lyme disease can be kind of a chronic infection because it keeps changing its surface proteins. Our antibodies aren't any good, and the antibodies are what help our phagocytes engulf the organism. There's other infections like gonorrhea that are really good at that too. So another mechanism there. Okay, and lastly, as we finish here, once the cell has to be able to go out, or the pathogen to infect other organisms. So portals of exit are the same places that it would have come in the first place. The rule of thumb is it's usually being shed in much larger numbers than what it came in. So things like coughing, sneezing, breathing, they're leaving, those pathogens are leaving on our saliva. When there's a fecal exit, they're leaving in huge numbers of pathogens can leave in diarrhea. I just love this picture. If you've never seen a cholera cot, people that are ill with cholera, they just lay on these cots and the diarrhea just flows out of them. It's just a nasty, nasty thing, but you can imagine how pathogens can leave in huge numbers there. Less commonly through the urine here, but certainly sexually transmitted diseases are leaving through semen or vaginal discharge, and even through blood. Okay, so good. We got through chapter 14. That worked out just about right. I know our time's up. The exam will cover through chapter 14, and I will push chapter 15 homework off to be due later. So have a good break.